Good morning, afternoon, evening, everyone. Thank you once again for joining us for the Center for Microbiome Sciences webinar series. This week, I am presenting again, but this time on ecology and ecological statistics. This is sort of an intro, but this is kind of required background for once you've done the data processing, what to do with the actual data that you've collected. So, all right, as with last week, if you have questions, there's a Q&A option available. So feel free to use that throughout. I will stop when I can if necessary. Otherwise, I will mostly wait till the end to answer questions. Okay, all right. And if there's any issues with volume or what you can see or what you can't see, feel free to let me know that as well. All right. So just a reminder of the webinar, where we're at and where we're going. So last week I presented on high performance computing, so using Unix and Linux-based systems in order to process your data, but kind of getting comfortable in that space. The lecture was recorded, so if you weren't able to attend last week, the recording will be uploaded and everyone will be notified when that is available. This week, we are doing, as I said, ecological statistics. Last year, we also went through using CHIME and Mother to analyze amplicon sequencing data. We decided this year, since that doesn't change too much, is to go ahead and focus mainly on the ecological statistics part of that. Last year's lectures and materials are still up on the COMS website and available for anyone. As I said, this is sort of an introductory treatment to ecological statistics. It is a massive field and constantly evolving. So this is meant to be sort of a background and basics to get everyone started. Next week, we will have a lecture from Ahmed Zaid, who will be talking more about ecological modeling and advanced statistics. All right, get started. So before we start talking about statistics and analytical methods, we're going to mention a little bit about community ecology, sort of the why are we doing any of this? Okay, my apologies. I'm going to adjust my microphone here. Give me a second. Okay. All right. All right, hopefully that is a little bit better. I've adjusted the volume upward a bit. Okay. Okay, so why are we doing any of this? What is the point of this course is titled Microbiome Informatics, but the microbiome part is, of course, where we start, right? And so we study the microbiome in an ecological framework. And so community ecology then is, as has been defined, to gain an integrative understanding of how biotic and abiotic factors mold local species pools at different spatiotemporal scales. So we have communities of interest, whether that is with oceans or soils or human-associated microbiomes, and understanding how those communities are formed, what alters their formation, what shifts their overall structure and interaction with each other, and how they interact with other organisms outside of themselves. So one of the things that is very of great interest to people are assembly processes. Right? How do we start? How do communities come together? How do they form? And this is of great importance to people who study also perturbations. So if something happens to a community, if there's some sort of ecological disaster, or some sort of insult to the community, how does the community reform? How is the community resilience and resistance to change and external factors? And just these are some of the key questions, and this is not microbiome specific, but how are species related to habitat characteristics? So in my field of oral microbiome research, we see clear and substantial differences between microbes that live in the saliva versus microbes that live on the surface of the teeth above the gum line, and even microbes that live on the surface of the teeth below the gum line. And so each of those different biomes has different characteristics that limit and filter what organisms can survive there. And so this is a good quote. I think that it's important for everyone to keep in mind. So that the thing that we need to keep in mind is understanding the linkages between what's happening at the level of the operation of the organisms themselves, so how they behave, their internal physiology, and external properties outside of the individual organisms, such as productivity and resiliency. Right, so these are, we focus on bacterial species a lot, but other people have called, for example, the microbiome a superorganism. Right, they communicate with each other, and so there's multiple levels to look at, from the organism level to the community level, to the interaction with the external ecosystem. So we can look at in microbial community ecology, similar but more focused or somewhat different aspects of these things that we've discussed. So again, how the microbial communities interact with each other and with the outside environment, functional pathways, so we look at the differences between organisms, but also the difference between for example, gene functions and perhaps other proteins, different levels, metabolites. And so some of these emergent properties that we saw in the quote from the previous slide are taxonomic diversity, functional diversity and redundancy, stability, resistance and resilience, and community genetic potential. And so then how the question becomes, if we're interested in these properties, then how do we assess the properties? And so there are quantitative and qualitative measures. And so quantitative measures use some measure of abundance, whether that's, again, species counts, gene counts, concentrations of proteins or metabolites. In some cases, so in the oceans and soils type microbiome research, you might be looking at spatial differences. Qualitative measures typically use presence or absence, and these are most informative when you have communities that, as I mentioned with the oral microbiome, differ primarily by where they can live, so the saliva versus the teeth. And so there are benefits and drawbacks to both. And so typically people will use methods from both to obtain different views of their data. So one of the first things that we like to look at is, well, diversity, but there are different kinds of diversity. Alpha diversity was first defined by Robert Whittaker in 1960, and he defined it as the richness of a species, the richness in species of a particular stand or community or a given stratum or group of organisms. So language there isn't particularly clear, but we'll see what this means. So in microbial ecology, it is diversity within a single sample or set of replicates. So this is something that you can measure at the level of a single sample. So to be more precise, alpha diversity is richness and or evenness. So richness is essentially the number of species in a community. So we see here in the slide, in the community to the right, community to the left, community 1, we have 4 different types of trees, and so the richness index would be 4. And community 2, we have a richness index of 2 because there are only 2 different types of trees. And so because of this, we say that community 1 is richer or has greater richness than community 2. And so the way we calculate this differs. There are a number of different ways to calculate, a number of different methods people have developed to calculate richness. We can just simply count the number of species that we obtain from our method of inquiry. Of course, this causes problems, and some of the community ecological methods come from instead of microbial ecology, they come from, we'll say, macroecology, right? Going out to a forest and counting the number of bears and deer and that sort of thing. And with those, depending on the size of the area you're looking at, you can be sure that you've actually sampled everything or close to everything. With microbes, we have a problem where one, we're taking limited samples from some environment, and then we're taking those samples and breaking up the cells, extracting DNA or RNA, and then we're taking that and typically fractionating it, and so we're only grabbing a certain portion of the DNA or RNA, and then we're taking that and we're running it on some sort of machine to count, but those machines have inherent limitations in how many sequences they can read, and so at each step we are losing information. And so what you'll see with more modern approaches, but even sometimes with some of the older approaches, because you can't count all the bears sometimes, is that there's some estimation for the things that you can't find. So as I said, alpha diversity is richness and evenness together. And so again, we have community 1 has equal ratios of each of the 4 types of trees. Community 2 has the same amount of richness, so again, 4 different species, but those species occur in different ratios. So tree A is 80% of the trees, whereas tree B and C account for only 5%. And so community 1 then is more even than community 2. So this is accounted for in Pillow's index, as seen calculated here. Shannon's index is one of the more commonly used in ecology and even microbial ecology. It has some issues as others do, but it's one of its benefits is it's very easy to interpret and very easy to calculate. So the P here in the equation are proportions or relative abundance. So the for each sample, for each sample, you are summing over the total number of observations in that sample, and you're summing over the relative abundances. So the count for organism 1 divided by the total count of all organisms for that species, for that sample. And so you are summing up the relative abundance multiplied by the natural log of the relative abundance in order to moderate organisms or observations that occur at very high frequencies. Faith's phylogenetic diversity is the only measure of alpha diversity I know of that incorporates a phylogenetic information. And so this generally gives you the sum of the branch length covered by a sample. So from the tree root to the tree leaves, if you take a sample and follow it out, how much of the tree is sort of covered by each of those samples. So we can compare alpha diversity, and we'll get to the statistics of this later. These are simple box plots, and this figure is actually produced by QIIME2. So you can go back and you can use QIIME2 to compare these statistically. And so alpha diversity is interesting, but it only gives you a very limited view of what's going on, because you're only looking at, you're not comparing samples you're not directly comparing samples to each other. You're only looking at individual samples, then comparing them with univariate statistics. And so what are the other sort of patterns of interest that we can look at in ecology? And one of the ways that we do that is look at what drives these patterns that we're interested in. And so we'll go through each of these. And so the exploratory methods look at relationships between samples based on their makeup, right? How many organisms of each type do they have? How many whatever of some observation? And so one of the things we can look at is community dissimilarity, right? We want to know, for example, again, going back to human health, because that's my area. We want to know, we want to know how we sample subjects, healthy subjects, and we sample disease subjects. Right? We sample the same location in each subject, so take saliva samples or subgingival microbiome samples, and we want to compare those communities that are essentially in the same locations, but have slightly different or have differing properties of those environments. Right? So in health, you have one set of properties. In disease, you've got lots of inflammatory processes going on, different oxygen gradients, that sort of thing. And so we want to be able to compare between these two communities, between health, between disease. And we can compare multiple communities in multiple ways. But one of the most common ways to do this, and we'll get to the name for this is also called beta diversity, but we'll calculate a pairwise similarity or dissimilarity between all of the subjects. So for a dissimilarity matrix, you're using a measure of dissimilarity. So this is on a scale from 0 to 1, 0 being samples are identical, and so you'll see that kind of diagonal across the matrix there, where all the samples compared to themselves have a 0 dissimilarity. And if you look at, for example, the highlighted section, you go from 0.58 down to 0.82, and so those samples are all relatively dissimilar to that first the first sample in the column there. So there are many different measures of community dissimilarity. These are some of the most common. The first 3 were developed originally for standard community ecology, so Jacquard, Sorensen, Bray-Curtis. Jacquard is presence absence, so it looks at whether samples are there, whether in each sample. To back up, so the dissimilarity matrix is calculated by taking each sample and representing each sample by a list of, in the case of bacterial species, for example, each sample is represented by a list of abundances. And so you go through and say, okay, organism A, this amount, organism B, this amount, organism C, this amount. So you have a giant list or a vector of these counts for each subject. And then for each subject, you go and compare subject A to subject B, subject A to subject C, and you do these pairwise comparisons using the measure or metric of similarity or dissimilarity. Now the nice thing is that any measure of similarity can be turned into a measure of dissimilarity by simply subtracting from 1, which is nice. So you can turn a similarity metric into a dissimilarity metric by 1 minus the similarity metric. So Jacquard simply looks at how the Jacquard calculates similarity or dissimilarity based on essentially the number of shared species, right? So if a sample has all the same species present and none of the same species absent, then those 2 samples are identical. Bray-Curtis is one of the more often used quantitative measures because it's somewhat relative. It's relatively insensitive to outliers. Unifrac is the only one on this list that was designed for microbial ecology, and it takes into account, similar to the Faith's phylogenetic alpha diversity, Unifrac takes into account the phylogenetic relationships among the species between 2 samples. So and again, the Jacquard distance, as I described, and Bray-Curtis similarity also looks somewhat at the difference of the ratio of shared and non-shared species. And this is the unweighted version of the Unifrac distance. And so this is a qualitative. This is more similar to Jacquard, but it's again similar to Faith's phylogenetic diversity. It's defined as the fraction of the branch length that leads to descendants from each single community, but not from both communities. And so we see here in the example, we're going to have different layouts, different participation from the blue group versus the red group. On the leftmost side, we have perfect equality. On the rightmost side, they're about even, and on the left side, on the right side, they're completely disjoint. And so that's the range. So again, given that it's a similarity metric, it ranges from 0 to 1. There are different versions of this. So the weighted version of Unifrac incorporates even more phylogenetic information, incorporates branch lengths. So how different sort of the evolutionary distances between organisms. So for example, if you have a whole bunch of counts from one genus versus another sample that is composed of organisms from different classes, a whole bunch of different classes or families, are going to have different weighted unifrac distances. There's also a variance-adjusted weighted unifrac distance that looks to see if sequences are randomly distributed among the leaves of the tree. And there's a generalized unifrac difference that contains an extra alpha parameter controlling the weight on abundant lineages, so that the unifrac metric is not dominated by highly abundant lineages, similar in a way to the Shannon alpha diversity that we mentioned. But again, this is sort of an active area of research, so there are new papers coming out occasionally making tweaks and differences to these distances. Okay, let me answer a couple questions real quick. For the question about, there's a question, how are batch effects handled in sampling microbial environments over time due to all the processing that happens when sampling microbiomes? Yeah, so that is a good question, and it goes, part of it goes back to designing your experiments, and so especially if you have groups of interest, you might sample them all at different times. You might even sequence them at different times, but it's important to, if possible, design your sampling and sequencing strategies so that you're not, if you have group A and group B, if you can, design your experiment so that you're not just sampling all of group A and then sequencing all of group A and then sampling all of group B and then sequencing all of group B. If you can sort of mix them, especially if a lot of studies will collect all the samples and then send them out for sequencing, but they'll send them out for sequencing in different batches, hence batch effect. But if you can, if you're in that situation, then it's very important to randomize which samples you send out in each batch. So again, don't, if you have two groups, send out half of group A together with half of group B. And then in the second sequencing run, send out the other halves of each of the groups. And there's studies looking at batch effects from those that I mentioned, from different sampling times to, well, that's actually something else I forgot about too, is that depending on what you're sampling, even the time of day can affect what results you get. It's, like I said, handling batch effects starts with study design and all the way through to sequencing, how the sequencing is done, who does the sequencing. If you have, there can be batch effects between different machines, even if it's in the same lab, or different operators. You've got two different people operating the same machine. People have seen batch effects from those. So there's a lot of different things to take into account in terms of sample design and sample execution, or study design and study execution. But there are some methods for handling those. I'm not going to talk about any of those in this lecture, but there are methods for sort of, we'll call it post hoc or bioinformatic / statistical treatment of the data to remove batch effects after the fact. So that is possible, but it's always better to mitigate those up front if possible. And then a question, can unifrac be applied in shotgun sequencing data where species don't share a common ancestor? So, yeah, okay. So, when you're creating a phylogenetic tree, you don't necessarily need to have, one, you don't necessarily have to have a complete tree. Right? You don't need to have a complete tree of life every time you're calculating unifrac. So you can simply create a tree from the sequences that you have, and calculate branch lengths based on that. So in that case, when you're dealing with shotgun data, as long as you have some method of creating a tree, for example, some what's the word I'm looking for? Some gene, some, for example, single copy gene that you can actually align to get the tree. Then you can do this sort of thing. If you have, now if you're talking about different domains of life, for example, that could be a little more difficult in comparing them. But if you're comparing different domains of life, then, well, no, I guess, yeah, in shotgun sequencing, you can get viruses and bacteria from the same thing. But yeah, you wouldn't be calculating the diversity. You would, in those cases, for example, if you're looking at bacteria and viruses, you would be calculating. It would be better to calculate the diversity separately for those different domains of life, if it's not possible to have, as the question said, any sort of common ancestor. Is it possible to use diversity measures on batch effect corrected data at all? Considering they change the distribution of the data. Yeah. So it depends. So for alpha diversity, probably not. If you're dealing with transform data, mainly because alpha diversity requires counts. But for beta diversity, you can definitely calculate beta diversity on transform data sets, so that's not an issue. Okay. There's, in fact, I think I was planning on referencing it, and I didn't end up including it, but there's a good paper from 2014, I believe. Paul McMurdy and Susan Holmes. The paper is titled "Waste Not, Want Not: Why Verifying Microbiome Data is Inadmissible," something along those lines. But they go through and talk about differential. They talk about beta diversity, applying transforms as a method. Well, we'll get to this a little bit later, but essentially the answer is yes. You can apply transforms and normalizations to beta diversity metrics before calculating them. Okay. All right. So it's one thing to calculate these measures of community similarity and difference, but representing that data is also important. And so if you remember, I said that for measures of beta diversity, you are taking each sample and representing it as a list or a vector of organism counts or some sort of relative abundance or transformed abundance or normalized abundance, and comparing those vectors of abundances to all the other samples. The problem then is, though, that especially with microbiome data, those vectors that represent each sample are large, hundreds or potentially thousands, if you're dealing with gene data. And each of those, the number of things that you're measuring, because it's in a vector, is essentially the number of dimensions of your data. And since we can't visualize 5,000 dimensional data, we need some method of ordination to allow us to visualize those higher dimensional data sets. So this is a representation of the dissimilarity matrix. Each community is represented by a single plot point. And each method of ordination uses some form of dimensionality reduction to take your 500 dimensional data and collapse it down to 2 or 3, preferably, dimensions. And so depending on the method, it will give you some, it will often give you some measure of how much of the variance in the data or some measure of variation in the data is captured by each of those dimensions. So principal coordinates analysis is one of the more common methods for ordinating dissimilarity or beta diversity data. Again, here, as with all these ordination methods, two samples that are closer to each other have smaller dissimilarity values than those that have dissimilarity values that are larger. Now, principal coordinate analysis, some people mistakenly, because it has the initials PCA, this is not principal components analysis. This is principal coordinates analysis. Principal components analysis can't be applied to this kind of data because one, it can't really handle dissimilarity measures, and two, principal components analysis kind of assumes that the space you're dealing in is Euclidean-based space. And for most of the ecological beta diversity measures, that isn't the case. If you'll notice, the earlier on I mentioned whether or not the distances obeyed the triangle inequality. So the triangle inequality is just sort of a basic property of Euclidean space, that the shortest distance between 2 points is a straight line. And for the way that many beta diversity metrics are calculated, that's not the case. And so principal components analysis isn't the most appropriate method for analyzing these, for a method of ordination. So principal coordinates analysis can take dissimilarity measures. It doesn't rely on any particular definition of the space, but it is related to principal components analysis in that if you give principal coordinates analysis a dissimilarity matrix based on Euclidean distances, then it effectively becomes principal components analysis. In any measure, in principal coordinates analysis, and the next one we'll see, the important part is when you're scaling down from many multiple hundreds of dimensions to 2 or 3, is maintaining the distances between objects as you're shrinking dimensions. And so that's one of the things that PCOA can do as well. And so with the principal coordinate axes, you'll often see a number associated with them, and those are, so each axis has an eigenvalue, the magnitude of which indicates the amount of variation captured in that axis. And so you often see that percentage value indicating, again, similar to PCA, how much variation is captured by each of the axes. Non-metric multidimensional scaling, or NMDS, is another measure. Uses ranks instead of the original dissimilarity values that PCOA uses. And so NMDS is an iterative process. So it starts out at the highest number of dimensions, and at each iteration attempts to remove one of the dimensions, while at the same time minimizing the amount of shifting that the samples do. The relative distances between, it tries to keep the relative distances between the samples as similar as possible. And so one of the things you'll see, and on this graph here, the output of NMDS is a stress level. And that sort of generally gives you a general idea of how well NMDS was able to keep the relative distances between the samples the same as it reduced the dimensions down from n to 2 or 3. And so generally stress values below 0.15 are considered acceptable. Higher than that, and you can't really be confident that your two-dimensional plot is really representative of your original full-dimensional plot. And so people often ask, like, when should I use PCOA, when should I use NMDS? Generally, you're fine using either one, but in some cases, especially with many overlapping gradients, NMDS will often produce a clearer overview of the data distribution. And so for a real-life example, this is from a paper I published a couple of years ago, comparing people who don't smoke, people who smoke tobacco cigarettes, people who smoke e-cigarettes, people who used to smoke e-cigarettes, or people who used to smoke tobacco cigarettes and now smoke e-cigarettes, the former smokers, and then people who use both smokers and people who use both e-cigarettes and regular tobacco cigarettes. And so this is a beta diversity metric. This is actually using a third method that I didn't mention, linear discriminant analysis, as a method of ordination. But the idea is the same, that samples close together are more similar to each other in figure A in their organism counts, and figure B in their gene counts. And so we see here that the non-smokers and the tobacco smokers are very far apart, but that the e-cigarette users are highly similar to each other, regardless of whether they used to smoke tobacco or are currently smoking tobacco, indicating that as an environmental pressure, that e-cigarettes have some common pressure that are shifting the microbiomes when people use them to be more similar to each other, regardless of other exposures. Okay, so sort of an overview of the exploratory methods. The second thing we're going to look at are the interpretive methods. So these are constrained techniques that use some additional explanatory variable during analysis. So we can ask the question, does the microbial community cluster according to predefined sample groups? So we go back, and this is I realize what I said wasn't super clear, so let's go back here and put it more plainly. Okay, we've got this pretty picture here. Now, what do we do with it? How do we quantify these relationships, right? I mean, the blue group, or sorry, the red group and the green group look like they cluster together, but that's not enough. We need to really have a quantitative statistical measure for determining whether the groupings that we see in these ordinations of beta diversity are significantly different or not. Okay. Let me stop and answer one of the questions. So someone asked, which of the plots best reflects the biological reality? That's difficult to answer. Part of it is partly because the question depends on what measure of community similarity that you're using. And so each of the different methods of measuring community similarity or dissimilarity sort of answer different questions, right? Bray-Curtis, for example, measure is somewhat a measure of organismal overlap, right? It looks at shared versus non-shared species, whereas Unifrac, as we discussed, also looks at shared and unshared species, but through the lens of phylogeny, right? So both of those have aspects of biological truth, but they tell you different stories. Now, in terms of ordination methods, they're all to some degree correct. And to answer another question, the linear discriminant analysis often produces clearer separation because it looks at it utilizes between group distances, but ultimately any of these ordination methods are just visual representations. So there are pictures, but they're not quantitative metrics. And so the biological reality, to answer one of the questions, is really best answered by the statistical methods that we're getting at, combined with utilizing the proper, or utilizing measures of beta diversity that are important or tell you something interesting about the community that you're looking at. Right. So again, this is for this particular plot. These are both produced by the same ordination method, but A is looking at organisms, whereas B is looking at genes. So this is from a shotgun sequencing data set. And that's often what you'll see is, especially with gene sequencing, that there's a lot of similarity, right? I mean, all bacteria need some method of energy metabolism, right? Carbon utilization. So you're going to see typically, especially when you're comparing organisms that live in the same general environment, this is again subgingival bacteria, you're going to see a lot of overlap between functional data, gene data, that you might not see in the organismal data. But the methods used to produce these two plots were identical. Okay. So, okay. So we've looked at exploratory methods, so the visual representations, and now how do we quantitatively compare these? Or rather statistically compare these. So we need to come up with some statistical measure to test some hypothesis involving community comparisons. These are some of the questions that we can ask. So do the microbial communities cluster based on the groupings we've assigned them? So like I said, we've got the different users of e-cigarettes, for example. Is there a relationship between the distribution of the samples within the communities based on some underlying gradient? So for example, pH can often be used. Some of these, you can, in other words, it's sort of like in statistics, utilizing a covariate to see if that can help explain the distribution of the community members. Some of the other things we can look at are, and this isn't strictly limited to beta diversity, but these are some of the other questions we can ask around more statistically defined questions that we can ask around community ecology. Some of the tests we can do are univariate tests of significance. And so we kind of mentioned this before when I showed the CHIME plots, but we can compare, how do we compare alpha diversity between groups? As this really gets back to just basic statistics. Since alpha diversity is one number for each sample, you group all the samples, group all the samples by the groups of interest. and then apply standard univariate statistics. So those would be for parametric, which I just put up there for completeness, but in general for microbiome data, you're not going to have normal data, although there are ways around that. We'll get to that. So for most microbiome data, you're going to want to reach for non-parametric tests first. You lose power, but non-parametric tests are generally more appropriate for microbiome type data. And so I mentioned these in order. So the t-test is a pairwise test. Wilcoxon's rank sum, again, is similar to it's the non-parametric version of the t-test. ANOVA, analysis of variance, is a sort of group-wise test. It'll tell you whether there's at least, so for if you have more than two groups, if there's a significant difference between at least one of the groups. Kruskal-Wallis is the non-parametric version of that. And then Tukey's honestly significant difference test is a post hoc test that if the ANOVA test is significant, you can go back and check, do pairwise comparisons and see which of the groups are significantly different from each other. And for the non-parametric version of that, the Dwas-Steele test can be used. The other major form of univariate test that typically is done for microbiome data is differential abundance or differential expression analysis, depending on whether you're sampling DNA or RNA. And the basic hypothesis is that for each, so this is sort of a large-scale hypothesis testing. So you're running one statistical test for each organism or gene that you've measured. And so the basic hypothesis, or H naught, for each of the tests, there's no difference in average abundance for the organism or gene between the two groups that you're interested in. So a lot of approaches for this. Some of the earlier ones were developed for microarray data. So Lemma was originally designed for microarray data. It's been updated. But the two primary approaches for RNA-seq data, are developed originally for RNA-seq data, are DESeq2 and EDGE-R. Some people use those for microbiome data, but there have been some publications comparing those and finding that DESeq and EDGE-R don't necessarily handle. They're not the best at handling zeros, and so this basically goes back to the assumptions. For RNA-seq data sets, you typically have not that many zeros, but for microbiome data, your data set is often what's called zero inflated. So you have some higher proportion, 30, 40, 60% of your table is going to be made up of zeros. Now, that's not always the case, and there are ways to look at the level of zero inflation, and there are measures to mitigate zero inflation. And so some of those are taken into account in differential abundance analysis designed for microbiome data. NCOM-BC is one of those, ALDEX2, which interestingly was originally designed for RNA-seq, but it turns out it's not that great at RNA-seq, but it's a lot better at microbiome data. And an approach implemented in CHIME is NICE plus Phylar. And Phylar is a phylogenetic transform of the data. And we'll talk about that a little bit later. And so again, this is just a real-world example of differential abundance analysis. This is from another shotgun sequencing study that we did of gum disease. And so you'll see we have three groups. We have the healthy subjects, or the healthy teeth. We have healthy teeth in people with chronic periodontal disease. And then we have teeth in people with chronic periodontal disease, but with active disease. And so you'll see that the primary sort of outcome, there are two primary outcomes from a differential abundance analysis is a log fold change and a p-value indicating the whether or not that the assumption, the original null hypothesis, was valid or not. And again, it was whether or not each particular organism or gene had differences in mean abundances between the groups. And so this is called a Bland-Altman plot. And it's characterized by plotting on the x-axis mean abundance and plotting on the y-axis log fold difference. And so the log fold difference ranges from negative to positive, with the center being 0. And this is typically what you'll see in these differential abundance analyses, is that they use a method of encoding the log fold change to indicate whether it was up higher abundance in one group or lower abundance in one group. And so the log fold changes that are positive are up abundant, upregulated in the control group, and those that have a negative log fold difference are downregulated or less abundant as compared to the control group. And here we just have the ones that were significant bolded, and those with the colored diamonds around them were also genes that were in the core microbiome. So they were present in at least 80% of the samples in that group. Okay. One of the cool things you can do, and this is a visualization method that I developed, is applying hierarchical circle packing from the results of differential abundance analysis. And this utilizes, I want to say, this is from the seed database. And so the seed database is a hierarchical database. And so this allows you to visualize within each of those hierarchies, so you can see carbohydrates. And so within carbohydrates, you can see those genes that are associated with processing carbohydrates or virulence or DNA metabolism. And then within each of those circles, they represent genes that deal with those things. And those genes that are colored red are more significant. In this case, this is back to smoking. So we exposed biofilms to tobacco smoke and then measured gene expression. So just something else you can do with a visual method for interpreting the results of differential abundance analysis. So going back to tests of significance for beta diversity, as I had mentioned a while back, there are 3 primary methods that people use. Permanova is the most recent, and at least from reading the literature, one of the more common methods. But ANOSIM is a test for significant difference between, so all of these give you, so ANOSIM and Permanova both give you tests between object classes. The Mantle test has some different requirements, but essentially all of these allow you to test for differences in groupings based on beta diversity. And so it's important to know what hypothesis each of these are testing, as they are different, and so using them can give you different answers, or can give you different interpretations of your data. So ANOSIM, the null hypothesis is that the average of the ranks of within group distances is greater than or equal to the average of the ranks of between group distances. PermaNova is more clustering focused, so it takes all of the subjects within each group, measures the centroids for those groups, or the if you take all of your samples within one group and find the sort of center of mass of all those points, that's the centroid, and PermaNova goes and compares whether or not the centroids are whether or not the null hypothesis is that the centroids of each of the groups are the same, more or less, are equivalent between groups. And so if the centroids for each of those groups are different, then you'll end up with a significant result. And as I said, PermaNova is nice because of the three of these. PermaNova can be used as a group-wise comparison. So PermaNova stands for permutational ANOVA. And so as with the original ANOVA, it is a group-wise test, but it can be also used as a post hoc test. So if you get a significant PermaNova result telling you that at least one of the groups is different between all the other groups, then you can go back and run PermaNova in a pairwise manner between all of the groups to give you which specific pairs of groups are different from each other. And this is something that you can do both in the R package vegan and QIIME will also, when you do tests of beta significance, QIIME will give you an option whether or not you want to do a pairwise test. And the final mantle test, the null hypothesis for that is that there's no relationship between the interpoint distances in one distance matrix and interpoint distances in a second matrix. So the mantle test is somewhat different, where it's really looking at comparing two different distance matrices instead of looking at the groups within one distance matrix. So the mantle test will often be used to look at whether there are differences in environmental gradients, for example. So that the pH example I mentioned earlier is one of the ways that the mantle test will be used. And so this is just sort of an overview of ANISIM. So as I mentioned, it looks at given a dissimilarity matrix with defined groups, that it ranks those dissimilarities. And the ANISIM statistic compares the average of the ranked dissimilarities between the groups to the average of the ranked dissimilarities within the groups. And so then you get an R value from this, and R value close to 1 suggests that dissimilarity between groups, suggests dissimilarity between groups, whereas an R value below 0 suggests that the dissimilarities are greater within the groups than between the groups. And as with many other of these non-parametric tests, significance is determined by a permutation. One other thing I want to mention, canonical correlation analysis is a useful way to look at, as I mentioned before, how environmental aspects correlate to the microbiome measures that you're looking at. Now, I will say canonical correlation analysis in its basic statistical form isn't entirely appropriate for the assumptions of microbiome data, but there are more modern methods that are derived originally from CCA that can be applied. But this is essentially a way of, like I said, sort of correlating your environmental measurements with your microbiome data. And this is a more modern version. As I mentioned, this is from the R package Mixomics. This is the results of sparse projection to latent structures, PLS, visualized using a correlation circle plot. And so here, this is a nice method for comparing two different sources of measurements, similar to CCA, where you have the angle between two different measurements, so the blue and the orange. If the angle from the center is acute, then those two things are highly correlated, or are more correlated to each other. And the strength of the correlation is the distance of those from the center. And so anything sort of outside that unit circle, the inner unit circle, has a somewhat strong correlation. And so things, as I said, that have an acute angle are highly correlated. Things that have an obtuse angle are anti-correlated, and two measurements that are sort of at right angles are have no correlation. Okay. And so getting back to what drives patterns of interest, the next method is our discriminant analyses. And so the goal there is to define functions or find hyperplanes that will maximize the separation of objects among the different classes. And so this is kind of looking at so in exploratory methods, we've looked at are there differences? The interpretive methods, we've looked at sort of defining quantitatively and statistically if those differences are significant. And the discriminatory methods in general are looking at what drives those differences, right? So what organisms are different between groups, right? How those patterns of interest, or what specifics are driving those patterns of interest. And so one of the more commonly used in microbiome data, even though it's a very old method, it tends to be still pretty good. And they're recent, as you see, 2019 paper looking at random forest classification. And so the basic building block of a random forest classifier is a decision tree. And a decision tree attempts to, and this is a form of an early form of machine learning. And a decision tree attempts to take a data set with known classes. You split up your data into a training set and a test set. And with the training set, the algorithm attempts to learn some sort of rules to classify the samples correctly. Now, with random forests, you're taking those decision trees and combining many decision trees together to essentially form a consensus classifier. So the idea behind this, and you'll see this a lot with various machine learning methods, is the general principle that multiple weak classifiers combined can outperform one sort of giant monolithic classifier. And so with random forest, each of those decision trees is limited. So each tree is limited to a certain subset of features. And this is more machine learning terminology, but in microbiome, in terms of microbiome data, a feature is the thing that you're measuring. So whether that's organism counts or gene counts or whatever, those are the features. And so with random forest, it generates a number of trees, say 1,000 trees, and each of those 1,000 trees gets a small subset, 5, 10, or fewer, of the total set of features. And then the algorithm builds a decision tree, a classification tree, based on that small subset of features. And then it classifies all of the samples based on their labels. And you get some classification percentage for each of the trees. And so then once it has created all of those trees, and again, these are weak classifiers because they only have access to a limited amount of data, It combines the trees with some method. So one of the examples here is majority voting. So each of the trees is given a test. So we've gone through all the training data, and now it's given, each tree is presented with an example of one of the test data sets, and is asked to classify it. And if 60% of the trees say that this is class A, then that sample is graded as class A. So this is sort of the general background behind random forest. So again, the general principle is that weak classifiers combined together do better than one monolithic classifier, mainly because too much information can be too restrictive. So, and overall, random forests provides very robust predictions. Although I will say that, as with many of these methods, garbage in, garbage out is an applicable principle. And so there are studies looking at various methods of feature selection. So essentially pruning the number of measurement types that you give to the algorithm. So eliminating organisms or genes that don't meet some criteria. So for example, organisms or genes that have no variation, right? They're just across all samples, across all groups, they're the same, right? So those don't really give you much information to work with, so we can throw those away. And there are different methods of variable selection, or sometimes called feature selection. But the idea is to pre-process the data to remove features that are essentially not of interest, that you don't think will be useful in classification tasks. Okay. One of the other very often used techniques are graph or network construction. And so here we see a network composed, so a network is composed of, a network or a graph is composed of two things: vertices or nodes and edges. So the vertices are the circles, and the edges are the lines connecting the vertices or the nodes. And so one of the most basic measures of a network is a node degree. And here we have, so the node degree is defined as the number of edges that are incident or attached to a node. And this is an example of an undirected graph. You can also have directed graphs where those edges have arrows. And so the direction of the relationship between two nodes is important. So in a directed graph, the degree is defined as the number of incoming nodes as opposed to the number of outgoing nodes. But in an undirected graph, it's simply the number of edges that are attached to any one node. And at a basic level, this can give you, again, depending on how the network is constructed, this can give you, node degree can give you an idea of the importance of that node, right? If you have a node that has 500 things connected to it, it could be more important than a thing that has 2 or 3 other nodes connected to it. But again, this entirely depends on how you actually construct the network. And so, speaking of which, constructing the network, there are different ways to do that. And these are generally split up into association measures on the one hand. And so these typically are correlation related. And so, for example, you can create a basic network by simple correlation metrics. So for each of the variables, you calculate a pairwise correlation score, and you'll have typically some cutoff for correlation and or significance. So if you have, again, correlation typically runs from -1 to +1. So a correlation score of 0 indicates no correlation. and a correlation of -1 indicates a negative correlation. So one variable measured goes up while the other one goes down. So for example, in terms of organisms, you can have one species of bacteria that when compared to each other, one goes up while the other goes down, and then positive correlation, meaning that both go up together. There are issues with just using simple correlation, especially for microbiome data. But some of the measures that are metrics that have been used to deal with that are the bi-weight mid correlation, which is used by the WGCNA package, which is, I always forget what this stands for, weighted genome correlation network analysis. Yeah. But this will be one of the topics of Ahmed's talk next week in his advanced ecological statistics lecture. You talk slower. So the bi-weight mid correlation is median based as opposed to mean based and is less sensitive to outliers. SparseCC and Speakeasy were one of the earlier methods for network construction that were specifically designed for microbiome data. They take into account the fact that microbiome data is compositional, which we will talk about a little bit later. I put them together because they're more or less the same. They have some small differences. Mainly, SparseCC uses a log ratio transform, whereas Speaky uses a centered log ratio transform. Spring is a more recent one. I have a little description there, but it's a lot more complicated, but you can look up the paper if you're interested. But one of the things that Spring was specifically designed for was to handle zero inflated data, which, as I mentioned, is often common in microbiome data, but it's not always the case. So you can't just assume that your microbiome data is zero inflated. In fact, there's a paper I just remembered. I don't remember the actual title, but it's something along the lines of, is your microbiome data really zero inflated or not? So it's not always a useful assumption. So as opposed to association measures, networks can also be constructed by dissimilarity measures. And so this really goes back. You can use any of the measures we use for calculating beta diversity, because then again, you're calculating pairwise associations or pairwise similarities, which is what a network is. It's so your nodes again are OTUs, for example, and you've calculated your beta diversity metrics. Or sorry, your nodes are samples, and you're calculating beta diversity, and so two samples are have an edge connected to each other if their dissimilarity is below some cutoff, for example, or above if you're using a measure of similarity. And so this is an example of a Jacquard dissimilarity network. So this is derived. So first of all, this is created by thresholding the Jacquard dissimilarity matrix. This was created in phyloseq, in a very useful R library for microbiome analysis. But again, you can see here that not all the nodes are connected, because again, we're using some cutoff for connecting two nodes with an edge. So if you're going to do microbiome network analysis, I would highly suggest this R package that just recently came out, mainly because it sort of condenses all these a lot of different things together into one easy to use package instead of going back and forth. Especially some of the older ones like SparseCC have issues with speed, for example. There was a publication that came out several years later called FastSpar that was a C++ implementation instead of a Python implementation of the original SparseCC. But again, all these different methods are located in different places and have different levels of ease to run them or implement them, and this netcome package puts everything in one location within a similar framework. It's very useful for that aspect. This is the GitHub page. You can go download it. Right. So the other thing I wanted to say about netcome is that so it implements 10 methods of association for network construction, 7 dissimilarity measures for network construction. It has 4 methods for zero replacement. So some of these, so as I mentioned, the like sparse-cc and speakeasy use log transforms. Well, you can't apply a log function to a 0. So all these methods that use log transforms have to use some method for replacing the zeros with something that can be log transformed. And so there are methods based on just adding a count. So some methods will just add a 1 to everything. And so all the zeros become 1, and then anything more than 0 gets 1 added to it as well. Or there's it's called a sort of a pseudo count that's added. Some people use something less than 1. Some very small, 0.001. There are more complicated methods that use Bayesian estimation to instead of just adding a single static number to everything, will add a pseudo count based on the properties of each of the samples. But anyway, so netcome implements 4 different methods for that, and then it implements 6 different methods for data normalization and data transformation, which we will go over. It also includes a whole bunch of methods. I think it's built on top of iGraph, which is a popular C-based or C++-based graph network library, and so it allows you to calculate a whole variety of metrics. So node degree is one that I mentioned, but there's betweenness, there's centrality methods, there's methods where you look for hub importance, all kinds of different stuff that you can calculate for structural qualities of the network, and then it allows you to calculate those and then do statistical testing between different networks. So for example, here on their GitHub page, they have 2 networks constructed from 2 different groups, so a warming group and a non-warming group, and then you can run various statistical tests to see if the networks of associations are different between the 2 groups. Okay, so there's a question, do dissimilarity measures used for generating networks give you the same interaction assumption problems as similarity networks? Okay, I'm not quite, if you could clarify what you mean by interaction assumption problem, I'll give another take at that. Okay. All right, so moving on. So we're sort of done with analysis. Well, we're not done with analysis methods, but I do want to mention methods for normalization and transformation. Part of the issue with any of these data analyses is, part of the problem in general with microbiome or omics data is that, as I mentioned earlier, one, you're not going to, it's impossible to count everything in an environment, and 2, even if you take 2 samples from the same place and then process them separately, just based on differences in processing and differences in sequencing, you can end up with different results, right? And so sort of like sequencing depth differences, for example. But so there are a lot of issues with counting and sequencing in terms of counts not necessarily representing the true underlying biology. Now, part of this is taken care of with the fact that we sample more than once, right? Or in other words, we have more than one sample. But in some cases, in general cases, you want to try to mitigate that as much as possible. So, and here this simple example kind of shows some of the issues. So if you look at figure A, on the top, you have an ecosystem. You're sampling from an ecosystem. And in the uppercase A, the figure on the top, your ecosystem consists of 8 total things. 4 of them are blue and 4 of them are red. And so you can see in the top box, you have your sample is consists of a half of each group is sampled to a fraction of a half, and so you end up with 2 of each, 2 of the blue, 2 of the red. But in B below, so 1, you have a much larger ecosystem. You have 6 of green, or 6 of red, and 6 of blue. And also, I'm realizing this is not my figure, but I just want to mention color blindness is an issue with some people, not me personally, but I always think of this when I see simple colors like this. So red, green is the most common, but anyway. But yeah, so the top group, or the blue group, in the ecosystem both have, and the bottom group both have 6. But after sampling, we have the same number of each of them from the top box to the bottom box. So we have, in the top box, we have 2 each of blue and 2 of red. And in the bottom box, we have 2 of blue and 2 of red. So even though we have the same number of measurements between them, those measurements represent different fractions of the true ecosystem. In figure B there, we have a different problem where you have a much larger ecosystem on the bottom than you have on the top. And then so while the proportions are different, you're really getting a different view of the actual ecosystem based on sampling effort, is sometimes called. And so there are a number of methods for dealing with this. Some of the earlier methods, one of the earliest methods was rarefaction. And this is essentially subsampling to a given depth. So if you have sample A that has 10,000 organisms and sample B that has 2,000 organisms, you sample at random from sample A, and you extract 2,000 organisms from sample A, or sorry, 2,000 counts. So this is in terms of microbiome data, you would do rarefaction at the sequence level. So you would take all of your sequences for sample A, the 10,000 sequences, and at random pick 2,000 of them. And then you'd do it again, and then you'd do it again. And sort of do a permutation based on that. But the general idea is that you take your smallest sample, or the smallest sample that you're willing to keep. So you might, if you have some samples that have 10 sequences and your largest samples are 10,000, you're probably not going to want to sample down to 10. But the basic idea is that subsampling to a given depth to basically make the statistics nice, so that you have even sampling depth across all the samples. There are issues with that. And that paper I mentioned earlier, Waste Not, Want Not, Why Verifying Microbiome Data is Admissible, says, hey, look, we've got modern statistics, we don't need to throw away information, right? We can use statistical techniques to handle those differences in sampling depth. And so from that, we get various methods for normalization, or essentially scaling. And so these attempt to eliminate biases resulting from unequal sampling fractions or differences in sampling depth, as I mentioned. And so cumulative sum scaling was one of the earlier ones that really showed a lot of success, and a lot of people still use it. There's the trim mean of M values that EdgeR uses. There's a relative log expression that DESeq2 uses. And I just mentioned Wrench because it's one of the more recent ones that uses more sophisticated statistical treatments of the data. So I don't have time to go through all of the pros and cons from each of these. So this is really just introducing you all to the topic and giving you some place to start for looking at what you might want to use. But these are sort of the general examples and general categories. So outside of normalization and scaling, there is the option of transforming the data, right? So instead of, normalization and scaling typically start with relative abundance and then divide relative abundance by some factor. One of the easiest ones is the TMM, the trim mean of M values. And so it literally finds a scaling factor or divides all the values by that, sort of force things to be equal or even. But as I said, there's another method. So, and it's based in what's called compositional data analysis. And so the issue is with any relative data, such as microbiome data, and even though you have counts, it's still relative for all the reasons that I mentioned before. So one, it's relative because you're not sampling 100% of your environment, and even if you were, you're not sequencing 100% of your environment. So it's at each stage of processing, you're losing information, and so really all you can, since you can't, if you have a species A and a count of 10, you can't say that there were exactly 10 organisms of species A found in that environment, right? You don't have that sort of ability to have an absolute count. So really what it is, when you have microbiome data, this kind of sampling data, what you really have is relative data. And so for example, if you'll notice, all of the comparisons that we've been doing are between groups, right? So we aren't looking directly at, so the beta diversity does do pairwise comparisons, but we're not making statements about the absolute abundance of any particular organism, mainly because of this issue. And so this is sort of an illustration of what's called the Aitchison complex, sort of one of the earliest realizations back in the 60s, I believe, where people started working with this kind of data. But the different proportions, so here A, B, C, and D are 5 different compositions, but because of the way proportions work, relative abundances work, A, B, and C are all equivalent, even though they might have different sets of proportions, and D and E are equivalent to each other. Another good way to think about this is, going back to the circles with the sampling, is with relative abundance, first of all, everything has to add up to 1, right? And so you're just limited by that. It's called the simplex. But if you think about it, if you take one organism that has a certain relative abundance and replace it with another organism that has a different relative abundance, right? You're still adding up to 1. But because you've added a new organism, you've swapped out organisms, all of the other relative abundances have to change because it's all relative to what you added. So if you have species A is 10, species B is 5, and species C is 1, right? So your total is 16. So your relative abundance for species A is 10 over 16. Your relative abundance for species B is 5 over 16. And then your relative abundance for species C is 1 over 16. But if I go ahead and replace species C with species D, which has a count of 20, my relative abundances all change. But what they add up to doesn't. They all still add up to 1. And so the argument for compositional data analysis is that instead of attempting to scale the data to sort of squeeze them to be more similar to each other, is that we simply transform the data to a different space. And so again, this is a whole wide field of mathematics, but they all involve some sort of log ratio transform. The most common by far, I will say, is the centered log ratio transform. Although it does have some issues. Again, not going to go into them. You can do your own searching for that, but the isometric log ratio transform was designed to resolve some of the drawbacks of the centered log ratio transform, but the isometric log ratio transform can be, because of its properties, can be difficult to calculate in the first place. But the main reason I mention that is that there's a microbiome specific log ratio transform that takes a phylogenetic tree into account. And if you remember before when I was talking about differential abundance analysis, the implementation of NICE in CHIME makes use of this phylogenetic isometric log ratio transform. So I will point out this is one of the earlier articles on, not the earliest, but definitely the most, what's the word I'm looking for? Opinionated, just from the title. And while they do make some good points, I also have some problems with some of the things that they say. So for example, one of the things they say is, well, we've got this log ratio transform that transforms the microbiome data into a Euclidean space. And so forget all of these methods of beta diversity that we talked about. Just apply a log ratio transform and then apply PCA, principal components analysis. And then let PCA tell you what's different. So there's several problems with that. One, and this is not something I have seen discussed anywhere in the literature, but one of the problems with that is that what do you do with the zeros, right? And so this is, I should have added a slide for this, but where do zeros come from in microbiome data? Well, they can either be because the species isn't there. They can be because the species is there, but is at such a low abundance that it was essentially missed in the survey that you did, the sequencing that you did. Or C, it could just be technical, and that even if it's something that was had a decent abundance, it was simply just missed either by your sampling or by the sequencer or whatever. And so various ecological metrics, so Bray-Curtis, for example, just ignores the zeros. There are other methods that attempt to make certain assumptions about what the zeros came from and deal with zeros that way. But if you're simply going to transform to Euclidean space and then use, as I said earlier, principal components analysis, implicitly assumes a Euclidean distance metric, right? So two things that are physically close to each other in space are more similar. But again, that goes back to the problem of zeros. If two samples are all zeros, then PCA is going to say those are identical, right? But again, we don't know where those zeros came from, right? Or even if they're, that's sort of an extreme example, but with principal components analysis and a Euclidean distance metric, any two samples that have shared zeros in a species, those zeros are going to bring them closer together on that dimension. And we can't necessarily say that that's the case because we don't necessarily know where those zeros came from. So that's one of my issues with just the transform it to Euclidean space to Euclidean space and run PCA. My second problem is one of interpretation, right? A lot of these ecological metrics, beta diversity metrics that we've been talking about, they have a specific goal in mind, right? They tell you something specific about how all the samples are related to each other, right? And so if you simply throw all that away, you're throwing away a lot of potentially useful interpretive information when you do that. So anyway, it is important to note that, like I said, even though the provocative nature of this title is very absolute, there are other criticisms of compositional data analyses. The compositional metrics themselves, as I said, the log ratio transforms themselves have some issues. And all this to say is that nothing is absolute. And that's unfortunate. I would love to be able to say, use this method all the time and you'll be great. But the truth is that, in different areas, so in network construction, for example, it seems pretty clear that network construction is made better, at least for microbiome data, with log ratio transform data. In other methods, there are, so for in differential abundance analysis, for example, all of the RNA-seq methods use some sort of normalization as opposed to a log ratio transform. And so there are some differential abundance analysis methods that use these compositional transforms, and there are some that use normalization or scaling methods. And the evidence was sort of unclear as to which is better all the time. So, but again, this is what happens when you're sort of in a relatively new field, or a field that is constantly creating new methods, is that you pick what you think is best for your data and you go with it. Okay, so let's see. Okay, well here we'll get it. So this is a question about data imputation. Before I'm going to finish up here with this data, or sorry, with my slides. And so one thing I wanted to mention, we didn't mention this last year, and really what I'd like to do is have an entire lecture on just multi-omic analysis, but I didn't want to have a lecture this year without mentioning it at all. So I'll just give a very quick treatment of multi-omic analysis, simply because it's becoming much more popular. Sequencing costs have come down, the costs of other biological measures have come down, and so now, whereas before, 10 years ago, it was super expensive to do even a 16S amplicon study. Now it's relatively cheap, or at least within the budget of most federal grants, we'll say, whether you're in the United States or in Europe or elsewhere, to do multiple sequencing measures, so or multiple molecular measurements. And so these methods are still developing. They're still, this is definitely at the sort of bleeding edge of science. But as I said, it's becoming more and more common, so I think it's important to mention. And so multi-omics is defined as analyzing simultaneously 2 or sometimes in some definitions 3 or more omics data sets measured on the same set of samples. And that's sort of the key point, is that for a multi-omic analysis, you have to have the same set of samples. Now you can have missing samples. Some of the omics can be missing some samples, but overall, the omics have to measure on the same set of samples, even if a few are missing here and there. Some definitions also require that the omics come from different layers of biological regulation. So, DNA, RNA, protein, metabolites, those are all from different layers of the biology. But a lot of these techniques don't really care how you apply or what you apply them. So, for example, you could technically have multi-omics if you're comparing the counts of bacteria to counts of viruses to some protein measurement or something. So, but the goal is to determine some mechanism of a biological process, to explore the effects or relationships from one omic measurement to another, to map biological pathways, to find biomarkers. Biomarkers are huge in human health-related fields, right? And in this case, multi-omic biomarkers. So what patterns, what multi-omic patterns, what bacterial species plus proteins plus metabolites all come together to form a predictive set of measurements for some disease state, for example. So, but this is all based on the assumption that more data is better, right? The more different ways we can look at some biological, at some ecology, the more information we can have, and the better we can analyze and understand the multifaceted aspects of whatever is happening. Okay. And so this is just sort of a general grouping of the different methods. So there's correlation or factor analyses. There are clustering or classification or prediction. These are typically more machine learning based rather than statistically based, which is the correlation or factor analysis types. There are multi-omic network construction and analysis, and these range from simply finding ways to connect different measurements together to more focused and what I mentioned, sort of the multi-layered. So actually finding almost sort of reconstructing biological pathways, right? So what organisms produce this protein that's involved in this pathway, that kind of thing. And that typically, all the methods I have seen to date that utilize that sort of network construction method have been focused on human data. So I've been looking for something, but it just tends to be fairly specific to human-associated data. There are autoencoders that are, it's a type of neural network for unsupervised learning, so it could be included under the clustering classification group, but it's different enough that it can be put on its own. And then there's batch effect correction. So this is the newest sort of area of multi-omic analysis, is how do you remove batch effects between different omics types. Since you're adding data, there are always drawbacks to that. So there are a lot of problems that have to be dealt with to do a multi-omic analysis. The first of which is sometimes called data harmonization. So each of the different omics types has a different underlying measures. So how do you normalize or scale? I wrote scaling twice. That clearly was meant to be something else, but I don't remember what it was at the time. I think it was transformation. It should be normalization, scaling, transformation. but how do you treat all the data sets so it makes sense to compare them? A lot of the methods will simply say each omic type should be normalized according to the assumptions for that omic type. And then once you've normalized each one individually, then you can bring them together. And that can be difficult for some areas. Like as we've already seen, even just for sequencing data, the question of normalization or transformation is not a solved issue. And that's true for proteomics, metabolomics, immunofluorescence. As we'll see, as you see in the list down below. But missing data across multiple types. I have this issue right now with some data. I have a study that has 16S data, it has metabolite data, and it has immunofluorescence data. If we're going to, so there's two approaches to that. So either A, you throw away all the samples that don't have measurements in each of the different measurement types. But depending on how that looks, you might be throwing away a lot of data. If you're only missing a few, if only a couple samples are missing a few of the omic types, then you can get away with that. But if you're throwing away half your samples, that's not great. But there are other methods that use different, either imputation, which someone asked about. I haven't forgotten about your question. Data imputation is a method for saying, okay, well, there's missing data here. Let me try to impute or guess what the value of it might be. So data imputation takes multiple, there are multiple ways to do data imputation, from the most simple of simply averaging, and so replacing any missing data with the average for that measurement type. So, if you have, let's say you're missing a count from, you're missing a metabolite from sample A. So metabolite A is missing in sample A. So you go through and measure all of the metabolites for the group that sample A is in, find the average metabolite concentration for that, and then you replace all of the missing ones with that average. There are other more complicated methods involving Bayesian estimation and that sort of thing. To answer the question, so what are your thoughts about imputing data when a single data point is missing, or how do you recommend handling missing data points? Again, there are different methods. So the best, in my opinion, it's better to use, if possible, statistical methods that don't require you to sort of make up data. That might just be a personal bias, but if it depends on the analysis methods, I'll say. So if you're using an analysis method that requires you to not have any missing data, then sure, imputation can be useful, but there's a limit. I'm not an expert on data imputation. I've used, there are several R libraries for doing data imputation that are nice, and they include a lot of these sophisticated methods that I mentioned. But just at a very broad level, it really depends on how much is missing. And so you need to, any data imputation method is going to be better or worse based on, one, how much data is missing, and two, how variable that measurement is, right? So if you're taking a measurement from something that has huge variability, well, any imputation you make is just essentially a random guess. But if you're dealing with a measurement that has limited variation, then any guess you might make might actually be pretty decent. But on the other hand, things that have large variability are often the ones that are important in discriminating between groups. So I tend to stay away from imputation unless it's absolutely necessary, right? So unless the method I'm using requires no missing data, and even then, it's if you're missing a whole bunch of samples from a certain measurement, it's probably better just to remove those samples than attempt to do imputation. Okay. But yeah, so going back to issues, so missing data across multiple types, there's a dependence between multiple omics types. Right? I mean, just sort of the biological dogma, DNA becomes RNA becomes protein. So there is some, a lot of statistical methods assume complete independence between measurements, which isn't always the case with different biological measures. And one of the biggest ones is dimensionality differences, right? So immunofluorescence assays might have tens of measurements. Organism counts could have hundreds, sometimes thousands. Metabolites, proteomics, again, hundreds or sometimes thousands. Genes, thousands or tens of thousands, depending on what you're sampling. All the way out to single cell seq now, which gives you literally millions of measurements. And so these are massive scale differences if you're trying to compare single cell seq with immunofluorescence assays, for example. And then just the more mundane, I've got there at the end, data wrangling. You're a lot of times you're sending these, you're preparing samples. You might have, even though the samples are from the same person, you might have a saliva sample, you might have a blood sample, and then each of those you might be splitting up into multiple subdivisions and sending out for sequencing, out for this, out for that. And so you're sending these out to different sequencing centers or labs, and then they're giving you data back. And so just the challenge of one, making sure you have all your samples named correctly, that you have all the samples, so just the basics of data handling, to make sure that when you're giving any of these multi-omics analysis methods data tables, that those data tables are in the same orders across all the samples, and all the samples are named correctly. So that's definitely something you need to keep track of and verify in your code as you're dealing with this sort of stuff. Okay, so like I said, I don't have time to go through multi-omics analysis is its own whole thing. I will recommend absolutely Mike Love, one of the authors behind DESeq. He maintains a GitHub page called Awesome Multiomics. It's one of these list GitHub pages, so anyone can contribute. And you'll see if you go to the pull requests, the general idea behind these is to just maintain a community list. And so anytime a new paper comes out, someone will put up a summary of it and issue a pull request, and then the repository authors will go and pull those pull requests, and then it will be added to this page. So this is all there is, just this one page. I've just taken a screenshot of this, but there's a giant list of methods all grouped up into, and actually I took this list from this page. That's how they group the multiomics analysis types. So this is constantly being updated and changed, so this is a very good place to sort of keep up with what's happening in the multiomics space. I do want to mention a couple specific methods, mainly because these may not be the best methods for your particular data set, but Diablo is part of Mixomics, which I mentioned earlier, which I like a lot because, one, it provides a whole bunch of different methods for just exploring your data, exploring the relationships between your samples and your groups, and kind of deconstructing what's important. But two, this and the next one we'll talk about are excellent examples of well-maintained code with excellent documentation, right? And I've gone through that list of awesome multiomics and said, oh wow, that method sounds really cool. And then I go to it, it was published 4 years ago. There haven't been any updates. There's no documentation. And I just literally, I'll spend an hour and can't figure out how to run it, right? So again, just a general comment. If I can't use your method, then it's not really helping anyone. So Diablo is a multi-omics analysis method. That's part of the Mixomics project, so mixomics.org. And its general goal is to identify correlated or co-expressed variables which explain the categorical or continuous. There is a modification to allow continuous outcomes of interest. It generalizes projection to latent structures for multiple matching data sets and sort of extends the sparse generalized canonical correlation analysis method. Its inputs are basically a list of data tables, one for each omic type, and a factor or a vector, a vector that lists for each sample what group they belong to, and then a design matrix indicating, if known, the relationship, how the omics types are related to each other. And it's sort of sort of vaguely defined, but generally getting at that idea that I mentioned of dependence between the omics types, right? Would you expect there to be some direct relationship between DNA and RNA? Yes. Would you expect a direct relationship between DNA and metabolites? Maybe, right? There is some relationship there. It's not like perhaps exactly direct, so you might scale that down, but there is some relationship. So the nice thing about this is, and I'll get to it, is that the next method I'm going to be talking about can actually help you figure that out. But the nice thing about Diablo is that it's, so one, it can help you analyze and pull apart your multiomics data and how the multiomics relate to each of the outcome, your group outcomes. But two, it is a statistical modeling method. And so whatever it learns from your data can actually be used on subsequent data. So if you run another experiment, you can use the Diablo outcome as a prediction measure, or as a prediction method. And so you can give it new data. So you can essentially, like a machine learning method, you can use the results of Diablo on your original data set to predict classes from new data types. And so this can be a useful method for data validation, for example, right? So if you do one study, run Diablo, come up with some set of results, and then you run a second study, and you run the Diablo predictor on the samples from the new study for a classification, you can see how good your predictions were, or how good your multi-omics measurements were for predicting those data types, or the group types. Multi-omics factor analysis is another one that I really want to go over. Again, this is, they have a great website. They have very good set of tutorials and documentation. But this analysis method is based on unsupervised statistical analysis. So you give it all your samples, but you don't necessarily give it the groups, right? You can, there's another method for that. But, its most basic, well, the MOFA method isn't interested in knowing ahead of time the groups that your samples belong to. And so, if you read their papers, they say that MOFA is sort of a very a generalized version of principal components analysis to multi-omics data. The idea is that MOFA infers an interpretable low-dimensional representation in terms of a few latent factors. So latent factors being some linear combinations, and so these are the purple, the W matrices that you see there, some linear combination of features that combine together sort of define that omic measurement. So one of the things you can do is, and like I said, this will give you the strength of relationships among the different omics factors. And so you can actually run, and I didn't come up with this data, or I didn't come up with this idea. If you, this is actually from a great YouTube lecture. I actually should have included it here, but you can, there aren't that many on multi-omics factor analysis, so if you search YouTube, it'll show up there. But the idea is that you run MOFA on all your data sets ahead of time, figure out the strength of relationship between them, and then take that information, go back into Diablo, and use the information from MOFA to create your design matrix before running Diablo. So, okay, so that was a whirlwind tour. Like I said, it deserves its own lecture. I will recommend, this is one of the more recent review articles on multi-omics. So I would definitely go there and definitely visit the awesome multi-omics list on GitHub for keeping up with that field. Okay, we're going to go ahead and stop here for now. I do have some R code I can run through real quick, but we are at 2 hours. So let's take a break here, and there is one outstanding question. So I'll go ahead and answer that, or I'll go ahead and talk about that. But you know, if anyone else, we'll take 5 minutes or so. If anyone else wants to answer a question, go ahead and ask your questions now. But we'll take a little bit of a break here. If you have to go, feel free to go. Like I said, I will go over after this some R code, sort of just as practical demonstration of some of the things that we've been talking about, how to actually do some of this. It's not going to be any new theoretical information, and new. It'll just be more practical stuff. So since we're at 2 hours, if you need to leave, feel free to leave. Again, this will all be recorded, so you can always catch up on it later. But I'm done with my slides, so feel free if you need to leave. But we'll take a few minutes, and then I'll answer some questions if there are any questions. And if not, then we'll just continue on to the examples in R, which are here. Okay, so there's a question. Do you recommend still using phylogenies, unifrac measures based on this recent paper? They mentioned a paper which I have not read, and it looks like it just came out last week. So I can't answer your question right now, but feel free to email me. I'll read through the paper, and if you email me, I'll hopefully have some sort of answer for you, but I'll need to read through that paper before I can answer your question. But I will put this article in the chat so that anyone else who wants to look at what they're talking about can see and take a look at that for themselves. Like I said, this is a brand new article, so. So the link for that article. Well, here, let me actually put the title first. The title as well. But basically, this looks like it's calling into question the use of phylogenetic trees. So. Okay. All right. So like I said, I'll have to read through that, but feel free to email me if you want some sort of if you want to have some sort of discussion on the paper, since I haven't seen it yet. All right, I'm not seeing any questions, so take another minute or so. Okay. Okay, so I got a question in R. How do you handle when an object is masked from a package? Do you mean when a method is masked from a package? So essentially, you can load different libraries will have different libraries might have the same method name, the same function name, and so when you load a library using the library command, actually I just want to make sure I'm not muted. Okay, good. Yeah, sorry. Okay, so yeah, so if you load multiple libraries, some of those libraries might have, yeah, okay, good. Okay, so right. So let's see. Actually, let me restart my R. Okay. If we go through and reload, let's see, packages. Okay, yeah, it looks like I reset it. But yeah, so to answer your question and to explain, as I said, some R libraries will have the same method names. And so when you use the library function here, you're essentially importing all of the function names from each of those libraries into your sort of global environment. And so if you load one library and then after it you load a second library that has a function that's named the same as a library you just loaded, you'll get a message saying function X in library A was masked by function X in library B. And so the way to deal with that is instead of relying on the global environment, is to call that function specifically. So for example, well here, let me go ahead and load all these libraries. Sometimes I think there might be an example of that. A lot of times what will happen is you'll see that libraries are actually re-implementing versions of base R functions. Okay, so it doesn't look like, okay. But let's say, okay, so let's scroll down here and look for, okay, so there's this estimate richness function, right? So if I say estimate, here, let me clear this real quick. Wait, no, wrong one. Oh. Okay, yeah, all right. So if I say estimate richness, there we go. Okay, so we can see here from the pop-up, this is RStudio, by the way, if anyone's not familiar. This is probably the best way to interact with R. But you can see here that estimate richness is a function in the fileseek library. And since I've loaded the fileseek library, I can just call the estimated richness function because it's part of my global environment. Now let's say there was some other library that also implemented an estimate richness function that as she said, as the person said, no, sorry, not she, as the person said, masked, get that message about a function masking another function, which means that when I call estimate richness, it will be calling the function from package B instead of the function from package A. And so, long story short, just call it directly by referencing the package first. So you can say fileseek, and you see RStudio will give you that option. So the double colon at the end of it will let you access any function in that library directly instead of relying on sort of the global namespace. So then I can go down and see estimate richness, and then I can call estimate richness specifically from the fileseek library regardless of what other functions are in the global function space. So this is the way you deal with masking, is just prepend the library name and a double colon so that your function reference is, so that R knows that you're trying to call the function from that particular library. Okay, good. All right, well, I haven't gotten a whole ton of questions, so anyone who is still here, thank you. So we will, as I mentioned, this code is up on GitHub. I showed the link a little bit earlier. Yeah, so we're just going to go through here some basic ecological statistical calculations. So alpha and beta diversity. There is an example of random forest, but the data behind this is actually from, was originally from Mother, Pat Schloss's, Pat Schloss, a professor at University of Michigan, his microbiome analysis package Mother, which we went over in the webinar last year. But yeah, actually here, that's a good idea. Let me put the link to the webinar, or the data in the chat. There we go. Okay. Okay. So I answered that question. Not sure if everyone can see it, but just in case not, I will put the link to this code in the chat. There are, I did run through the code this morning just to make sure things didn't change. There was one small change, but basically, let's see, what is it? The Adonis function in vegan is now, the original Adonis function is being deprecated in favor of Adonis 2. But other than that, the code hasn't changed from what's up on the webinar. So if you go to that link I sent everyone, under the ecostats folder is this R function, and then the RDS is the underlying data that I'll be accessing. As I said, this is from this is a mouse diet study from Pat Schloss's lab, where they fed the mice had different diets, high fat, low fat, etc. And they tracked these mice out over time, and there were male and female mice. So there's a lot of different ways that the data could be compared. And so I used this last year in the amplicon sequencing analysis section of the lecture to start from the raw data going to a final OTU table. So that all of that can be seen also in the GitHub link I sent everyone under the Data 2 or Chime or Mother, the process for analyzing that data, as well as specifically this was constructed by the Data 2. The RDS file was created by the Data 2 process that I went through. The Data 2 analyze.r under the Data 2 function folder in the GitHub page. But anyways, that's where the data comes from. So there's this mouse underscore ps underscore filt. So it's just the original mouse experiment data, but filtered. So that's where we'll start. So I've already loaded the libraries. These are the version numbers of each of those libraries that I'm using. And I believe, let's see, yeah. So I'm running R 4.2.0, which may or may not be the absolute most recent R. And it is not. I am a minor version behind. So the most recent version of R is 4.2.1. But I wouldn't expect any serious differences. The package versions are probably more important, but even then, a few versions don't usually change things massively, unless they're a major version number. You go from a 1 point to a 2 point. But anyway, so that's enough background. So we're going to load the mouse data here. In fact, and this is the mouse, the PS in the file name stands for phyloseq. So this is already a phyloseq object. Phyloseq is, as I mentioned earlier, an R library for handling phylogenetic data specifically. And so it actually comes with a very nice structure, as we'll see here. So if we go and look at what's in there, there is an OTU table. There's a taxonomy table. There is sample data, so this is essentially metadata. So data, all the information about each of the mice, their age, sex, diets, all kinds of stuff. This is essentially the sample data table inside of the phyloseq object is equivalent to the Chime metadata table, if you're more familiar with Chime. But essentially just a table of information about each sample, where each row is a sample, and then each column is some information about each of the samples. And then last, it has a phylogenetic tree embedded inside of it that you can use if you need it. Now, you can construct a phyloseq object with only one of these. So you can, if you just have an OTU table, no taxa, for example, you can still construct a phyloseq object, but it gives you the flexibility to incorporate all these different types of data into your analysis. So we've loaded the phyloseq object. And so the first thing we want to do is calculate alpha diversity. And so here I've grouped 4 of the alpha metrics into one vector that we will run simultaneously. So I now have a vector called alpha underscore metrics that contains observed, Shannon, Simpson, and Fisher, those metrics for calculating alpha diversity. So phyloseq contains, and again here if we, so you can see that plot richness is a phyloseq measure, so it kind of combines plotting with data, and so it actually takes a single or a vector and then plots each based on the grouping that you give it, in this case age, or binned age, since age is not a categorical variable. And so the plot richness function takes a grouping and 1 or more alpha diversity metrics and returns a ggplot2 object. Now this is important because they're using this line here is using a plus and this geom box plot. So it's important to know some things about how ggplot2 works. I'm not going to go into it, but it's if you're doing any visual exploration in our understanding how ggplot2 works is very useful. But essentially, plot richness returns a sort of a graph, sort of a data set, and geom box plot tells you that you want to visualize that data with box plots. So if we run this, and actually switch over to plots, let me run this, and okay, it's going to plot those, and okay. I'm not sure how this works over Zoom, but can people see this plot window that has popped up? If not, I will just adjust the frames inside of RStudio to, okay, good, thank you. All right, so I've been told that the plot window is visible. So, so you can see here, so it just produced box plots. Box plots are nice because they split everything up into quartiles, and then the line here indicates the average. And so just by quick visual, just by quickly visually taking a look at the data sets, you can see for you know, for Shannon, for example, the young mice, the young mice have a different mean than the middle-aged and the old mice. All of the boxes are overlapping. For observed, the older mice might be significant, but again, you know, that's just visual exploration. So I'll move that, close that for now. So some other ways. So this is just a little. The next set of code is just a little bit of we'll say visual sugar. So just exploring more of the different types of visualizations and how you can modify how things look. So let's run that. So this is going to create a violin plot, which is essentially a it's the idea is similar to a box plot, but it's essentially, whereas a box plot just shows you the ranges, the violin plot is essentially a histogram. It's a mirrored histogram. So we've replaced the boxes with mirrored histograms. So if you look at each of these sideways, it's a histogram distribution, or a kernel smoothed histogram of the underlying data distribution. And then on top of that, we've added a jitter scatter plot to the violins, so you can not only see the distribution with the violins, but actually see the data points themselves and how they're distributed. And so adding this sort of information on top of box plots is useful because sometimes the smoothing will kind of obscure aspects of the data. So actually looking at the data points in addition can be useful. And then the next one we will look at is, so we colored the violins by age, and now we will add the jitter, color the jittered samples within that, so we can kind of look at the different age distributions, because again these are binned, so we've got all of the ages from newborn mice to old mice. It was probably 2 years, I don't remember exactly, but let's say 2 years old. So say the young mice are from 0 to 6 months approximately. The middle-aged mice are 6 months to a year and a half, and the old mice are a year and a half to 2 years or something. But we've got a distribution of ages with age ranges within each of those categories. So here we've gone and added color based on the age factor to our jitter plot of the samples. So we can actually see here that there's no association, at least in alpha diversity and any of the alpha diversity metrics. There's no association between. That's not entirely true for Shannon. So it does look like in the younger mice in Shannon that there is some age dependency within the younger mice. So the younger mice might have some gradient in alpha diversity that doesn't seem to be present among the middle-aged and the older mice. So this, if you were looking at this, you might go back and you might either group the younger mice differently, or instead of grouping the mice into these artificial bins, you might actually use an alternate analysis method that, some sort of regression analysis method that doesn't require a categorical outcome, that can use a continuous or ordinal outcome. So, okay. And so the next thing we'll do is we've done a visual exploration of the alpha diversity, but we can use the estimate richness function, and then so the plot richness sort of skips a couple steps or combines several steps together. So it calculates richness and then immediately plots it. But if we want to actually do something, do some statistics with those richness measures that were plotted, we have to call the estimate richness, which is the plot richness uses the estimate richness sort of inside. But if we want that actual data, we can call the estimate richness. And so if we view the alphas table, so it gives us for each sample, for each alpha metric, it gives us the metric. So we can see for sample F3, so this is female number 3, day 0, female number 3, day 1, etc. So the observed richness is 222, the Shannon diversity is 4.07, etc., etc. And so this is the sort of underlying data table that was being plotted in each of the box and violin plots that we saw earlier. And so then we can go and run the Kruskal-Wallis test on these, which we can view it. Yeah. So gives us group-wise significance for each of them separately. So this doesn't give us pairwise significance. It just tells us that at least one of the 3 young, middle-aged, or old mice were different from one of the other ones. So in this case, there's at least one group significantly different from one of the other groups across all 4 of the alpha diversity metrics. And like I said, there are tests to go back, so the Steele DWAS, you go back and see exactly which ones, is it older versus middle or older versus young, which ones are actually significantly different from each other. Okay. So the next thing we're going to do is apply a log transformation. And we can see here, so this is one of the desirable qualities of a log transformation. This is the reason, one of the reasons, in addition to a log transforming the data into Euclidean space, which can be useful for some things, one of the major benefits to doing a log transform is that you can go from whatever kind of distribution your data has before to something that looks more or less normally distributed. And so you can take non-normally distributed data that you would need to run a parametric test on, for example, and after transformation, you might be able to run a parametric test on it and increase your power. So we're going to use this, and this goes back to one of the questions someone asked, but you can apply beta diversity metrics to transform data. So we will right now apply the weighted unifrac transforms. This is just from the distance function as part of phyloseq, and it takes multiple different methods. If we go look at the help for this, then the distance method should give us a list of, here we go, distance method list. So unifrac, weighted unifrac, double PCOA, is that Jacquard, Manhattan, Euclidean, yeah, so Bray-Curtis, Kolchinsky, Jacquard, Morcida, Horn. So there's a whole bunch of different methods that you can, distance methods that you can apply here that are part of phyloseq. So we're going to, for now, apply the weighted unifrac. So we'll calculate the distance matrix first, and then we will ordinate, so run principal coordinates analysis, also called multidimensional scaling as opposed to metric multidimensional scaling as opposed to non-metric, or NMDS, the non-metric multidimensional scaling. And then we can get the eigenvalues from the principal coordinates analysis for display on the plot, and then we can create an ordination plot with the plot points colored by the age binning again. Okay, and then we can zoom out and show that. The nice thing about phyloseq's ordination plot is that it doesn't show it square. So a lot of ordination plots you'll see, and I'm guilty of this as well, not really guilty, but it gives you a better idea, just based on the human visual system, it gives us a better idea of the relative contribution of each of the axes. So you can see here we don't have a square plot, we have a rectangular plot with the length of the axes based on the axis, the variation across the axis. So you can see here, so almost 50% of the variation in the data set is captured by axis 1, but only 10% of the variation is captured by axis 2. And so the literal physical size, oh, okay, that's odd. I wonder if it's because I closed it. Well, anyway, thank you for letting me know. I'll just expand this here for now. But you can see here that the plots, the ordination plots that phyloseq gives us are, the axes are actually scaled to match the variation captured by that axis. So it just gives us an additional little visual indicator of the importance of each of the axes. Okay, so let me bring this back down so we can see the data. Okay, apologies that the zoomed window seems to have stopped working. Yeah, actually, I'm going to stop sharing real quick, reshare. I wonder if it's because I was closing the window. Let's try that again. Let's see. Is that external window visible again? Okay. All right. Okay, well, we'll leave that alone for now. And continue going through the code. So we calculated the weighted unifrac and plotted it. So we'll go ahead and calculate the Bray-Curtis metric. Yeah, okay. Well, that's fine. Yeah, it's going to be difficult to bounce back and forth. And if I share my desktop, then everything gets super tiny. So I'll just go ahead and expand this window when necessary. So we'll go ahead and calculate the Bray-Curtis metric. Now you'll notice that it's sort of transparent, or sort of invisible in the background. You're giving it that phyloseq object to the distance metric, and the distance metric, since it's provided by the distance function, since it's provided by the phyloseq library, it knows what's inside of those phyloseq objects. And so here you'll notice for the weighted unifrac, we're not explicitly giving the phylogenetic tree, but it is part of that phyloseq object. If you didn't have a tree part of the phyloseq object, then it would complain at you. But you'll notice that there's essentially uniformity, so that distance function just takes the phyloseq object and a method. And so all we're doing to calculate Bray-Curtis is changing the method parameter. So we'll calculate that, PCOA, eigenvalues, ordination plot. And there we go. So we have the weighted unifrac, and we have the Bray-Curtis. And so there are differences. There are some differences in separation. The weighted unifrac shows a lot less group separation than the Bray-Curtis. So you might interpret that as there are fewer shared species between the young mice and the older to middle-aged mice. But by looking at the lack of separation for a lot of them, or the decrease in separation between the groups, you might interpret that as, and again, you can go back and actually look at what species are there, but you can interpret that as, since the weighted unifrac incorporates the phylogenetic tree, that a lot of these species that are shared among the young mice are species from the same part of the phylogenetic tree. So you might have a whole bunch of, in the young mice, you might have a whole bunch of species from the same genus, for example. And so to Bray-Curtis, those show up as different species, and so those push apart the samples between the young mice and the older mice. But since unifrac takes that relationship into account, the fact that you're only using a small portion of the phylogenetic tree, then it shrinks the distance, at least on the second axis, between those mice. So just an example of how different metrics, different beta diversity metrics can lead to different interpretations of your data. Okay, and then as I said, those are sort of visual explorations, but we actually want to do some statistics, so we can run the Permanova test with the Adonis function. There's a whole backstory about why it's called the Adonis function, but you can do a web search for that. But essentially here, and this follows the same sort of linear modeling approach that base R uses. So your input data is Bray-Curtis, the Bray-Curtis matrix, and your outcome is the binned age. So those are the groupings. So that's essentially how you run that. And then what we end up with is the output from Adonis. So it tells you ran a permutation test with 99 permutations, gives you the formula, and then it gives you the significance table. And so we can say, based on that, again, as with the alpha diversity, the group-wise test was significant. So there is some separation between, and remember the Adonis test, or sorry, the Permanova test looks at the null hypothesis is whether the centroids of each of the groups. And so here, let me just zoom in on this again. So the centroids, I kind of hand waved at it earlier, but the centroid is sort of the center of mass of all these points. So the red points are all the young mice, and so there are ways to calculate this, but generally you make up a point that kind of represent the center of all those points. And so the centroid of the young mice might be around here, and the centroid of the older, the middle-aged mice is probably up here, let's say, and the older ones aren't as many older ones, so it's harder to say, but it might be here. So we could pretty reliably guess that the groups that are different are young versus old and possibly young versus middle-aged, although some of the younger mice are up here as well. So given the size of the older group, that there might not be enough to say that there's significance there. But we can pretty much definitely say, without having to go back and do the pairwise comparisons, that it's probably the young mice versus the middle-aged mice that are significantly different. But anyway, so that's running the Permanova and getting the results there. So I don't want to spend too much more time because we're coming up on the third hour here. But I just want to go through, and again, I didn't come up with this. This is part of one of the PhyloSeq tutorials. I did modify their example to work with the mouse data, but the original is from one of the PhyloSeq tutorials. So this is loading Carrot and Random Forest. I don't actually remember why it's loading Magritter. Oh no, that's why. Yeah. So this, quick R lesson. So this operator here is part of the Magritter library, which is part of Tidyverse. But this is, I don't remember what the operator is called, but basically it says take the results from the previous function call, and I think it's called the pipe operator. Yeah, so take the results from the previous function call and pass it along to the next function call, such that the next function call will use the input from the previous call as the first parameter to the function. So that's what this, if you're confused, if you haven't seen that before, that's what that is. It's, instead of composing functions, you have kind of like nested functions going outward. This says let's turn that around and just call the first function and output it to to be the input to the second function, and the output of that can be inputted to the third function. And so it's just a different way of calling functions that can be a little bit easier to read. But essentially, we'll train a random forest library, and first thing is to split up the data into sample, into training and test data. So we've got our training mice and our testing mice. We're just randomly picking from the set of mice to be either in the training set or in the test set. And then we will run the random forest and plot the results here. And run this whole thing. Sorry, the random forest is still running. So it's constructing. So here we're doing it in two stages. We're training with the method random forest, and then we're running the predict. So we're testing. So we're first running random forest on the training data set, and then the random forest fit that we've discovered, we're then testing with the predict function on the testing data set. Okay, it's taking a little bit longer than I expected. But yeah, so and this is the same with any of these classification training methods, is that they're split up into training and testing. And so you'll train train a model. That's the word I was looking for. So with the training function, you create a model, and then you can apply that model with the predict function to the data set, to your test data set. And then basically the prediction, then with the prediction, it'll tell you how well because your test data set is also labeled, it'll tell you how well your model performed with here, what's called this down here, what's called the confusion table. So we've got two groups here. So this is instead of doing the three groups, we're just categorizing everything into young and not young mice. So I think that makes sense. I think this is days. So this is mice from 0 to 100 days, and then mice from 100 to 400 days. And so this is a standard table for classifiers. So it's this 2x2 table. It's called a confusion table. But basically it says, items on the downward diagonal were if I remember correctly, the column labels are the original class for those samples, and the row labels are the predicted label for that for those samples. So what this tells us is that 72 of the young mice samples were correctly predicted. So 72 of the samples that had the young mouse label were predicted as being a young mouse, and 2 of the samples that had an old mouse label were predicted as young. And on the flip side, one of the samples that was a young mouse was predicted to be an old mouse, but 45 of the older mice were predicted by the model to be older mice. And so you can see that's pretty good. Sometimes it'll list percentages as well and give you an accuracy percentage, that sort of thing. But we can just see from these numbers that it did pretty well separating them. And so we'll go ahead and plot that. Oh, I forgot to run that part first. And because I forgot to run that. There we go. So just a little lesson. 1, make sure you're running everything, but 2, it's good to know how to read error messages. So at first it said object RF procs not found, so I have to go look, oh, okay, I didn't run that line. So I had to go back and run that line. And then it complained that it couldn't find the function, that Magritter function, the pipe function that I mentioned. And then I realized, oh, I forgot to, even though I loaded the random forest and carrot libraries, I didn't load the Magritter library. So we loaded those, ran that line, we loaded the Magritter library, so we'll be able to run that line. That ran properly, and then we'll be able to run the ggplot. There we go. Yeah, I guess this was, it's not going to work. Yeah, okay. So, and I just did it again. Anyway, okay. So, yeah, this is just the output of that plot. Kind of rushing here. Okay, so we can also look at taxa importance using that. So this sort of the taxa importance is so one of the things you get with random forest is an importance score. One of the reasons that random forest is still so often used, I mean, 1, it does give you robust predictions, but 2, it gives you more information about what's actually important to the model. And so it'll give you a table. It'll give you a table that tells you, it'll give you a score for each predictor, each feature in the prediction. So it'll say, so in this case, the top 2 were Lachnospiraceae and Roseburia genera. Lachnospiraceae family and the Roseburia genus. But basically, you'll get this importance table where the random forest predictor will say, if we removed this feature from the predictor, the accuracy of the total model would go down by this percentage, or by this amount. And so you can kind of look at that in reverse and say, well, if I remove this predictor from the model, then my predictions will get 30% worse. So that means that that's super important. But if I look at this other one, if I remove this other predictor, then my overall model predictions will only get worse by 1%. So that's perhaps not as important. And so using that importance ranking really gives you the ability to drill down and see which species or genera or family are really important to predicting a particular group. Okay. And, oh, I didn't, let's see, I ran that. Okay. And then we can kind of plot that. Yeah. And so we can see, yeah, sort of that importance plot. So the abundance of discriminative bacteria between, in the top panel, the young mice, and the bottom panel, the lower mice. So in the, there are a few highly predictive, or well, one really highly predictive feature for the young mice, and a few lowly predictive ones. And in the older mice, there are a whole bunch of somewhat less strong predictors, but combined together, they provide discrimination. So that can also be, interesting to say, hey, there's a whole bunch of the community is characterized by a large number of less abundant species, whereas in the young mice, there's like one really big important OTU that's really just shifting the entire community away. So, okay, that is all I have. Oh yeah, I had intended to get into some mixomic analysis and didn't end up having time. Okay, so I will. That is it for today. So thank you everyone again for attending.